I'm Gilly, and I'm a comparative cognition researcher, and I work with great apes, and I work with infants, and I try to work out how we became upright, walking, talking, and tool-using great apes, both through evolution and through development. And what I'm going to talk to you about today is not yet published. It's not out there in journals yet because it's really new. It's translational work. And I think one of the issues with academia is it can be very, very slow to share information. And I think as researchers, as universities, we have an obligation to share it. So I'm bringing you this kind of research straight from the lab to you. And of course, things shift and change all the time, but I think this is going to be some really exciting new work. So stress and well-being in great apes. Maybe this works not this way? It was working. Is there an on-off? Oh, now it's working. Okay. So I'm just going to start with talking about well-being, because in humans, we've just kind of come to the realization that well-being isn't just about our physicality. It's not just about how physically well we are, but it's entangled with our psychological well-being as well. So if we were to consider how we measure, it's not going to work. All right, I might have to stand here. If we're to think about how we measure our life quality, how well we are, it's an absolute entangled mess between our physiological and our psychological well-being. And that's how we measure our quality of life. Now, if that's true for us, then what about other great ape species with whom we share over 98% of our DNA? And my supposition is that, of course, they also have this entwinement of their physiological and their psychological well-being. Now, we've spent many, 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 many years knowing that they're very closely related to us and actually using that knowledge to use them as models to understand ourselves better. So we've used great apes in medical studies, for disease, for all sorts of different kinds of models of the human body and mind so that we could improve our own lives. So this work is really an attempt to turn that paradigm on its head and use our understanding of our mental well-being and see if we can use that as a model to help great apes. Maybe if I push really hard, I can move away from now. No, not me. I feel like I haven't turned it on somewhere properly. No, okay. Now it's going in very quickly. That's not good either. Okay, maybe it's working now. Right, okay, so if we actually wanted to measure our well-being, we've all been to the GP, we've all kind of been for hospital tests, and we know that we can do things like take our blood pressure, get our blood work done, and this can give us a measure of our physiological well-being. And with our psychological well-being, still not going to work. We're going to do things like ask questions of you, right? So you might have a clinical psychologist or your GP asking you questions about how you're feeling emotionally, mentally, and we're going to make notes about that. And those two things together will dictate our well-being. Now think about a chimpanzee, a gorilla, or an orangutan. Now, taking your blood pressure or your bloods or your heart rate or your respiration might seem completely non-invasive as a tactic to understand the physiological state, but you're certainly not going to be able to go into a gorilla enclosure or out in the forest and ask a gorilla very kindly, could I please take your blood pressure, right? So it becomes a very invasive tactic. Likewise, psychological measures, we were doing these surveys, I can't ask a chimpanzee how they're feeling, right? They're not going to be able to tell me how they're feeling and what makes them stressed or fearful or anxious. So the way that we do measure ape well-being at the minute is that for physiological well-being, we're going to do things like look at their weight, look at their diet, look at their enclosure to see if we think it's big enough. You know, do they have good relationships, might tell us something about their psychological measures, and that's all going to be hearsay from the keepers. We might have some information about their stress hormones like cortisol from taking urine samples or fecal samples, and those will be kind of aggregate, and they won't be time locked to specific events. So they're not really great measures, but that's what we have, and that's what we currently use to understand the well-being of other great ape species. And so thinking about ourselves as a model to understand them, I was thinking, okay, what are the kinds of things that stress us? We generally get super stressed by social things, right? Social reputation. But we can also get stressed by cognitive things, right? So, for example, exams or solving difficult problems. And these are probably the biggest stressors for humans, right? There might be others. There might be, you know, something that jumps out at you while you're taking a walk and you're worried about your physical safety. But for our day-to-day life, it's generally social and cognitive stressors that we encounter. And to measure these things, again, we just use these physiological markers. You might look for the amount of sweat on your hands, so galvanic skin responses, but it's heart rate, it's respiration, it might be even looking at dilation of pupils and things like that. But there's another way, temperature, and this is where the translational work comes in. Temperature can actually give us some really interesting information about our arousal states that we hadn't really understood before, and this is all now coming to light. So using a thermal camera, so a thermal camera is going to look at radiation that's emitted by all objects in the world, and it changes it into a digital picture that is colored. And the color gives you the scale of the different temperatures. So, for example, if I shine my digital camera on my friend Ben, we get a lovely image like this. You can even see the video. The colors go from the darker colors at the bottom being quite cool, the reds and the whites at the top being the warmest ones. So can you see that there's a relatively even reddish color around Ben's face? The eyes are a bit hotter. That's not so surprising. We are primates and vision is our primary sensory modality. We do expect to see quite hot eyes, and we've got a lot of blood vessels around that area as well. With thermal imaging, we can take that image, and for every single pixel in that image, I can tell you what the temperature is. So each one of those little cells would give me the temperature of a pixel in that image, which is incredibly detailed information. And it means that we can take kind of regions of interest and look at the nose or the eyes or the cheeks or the chin or the forehead. And you might be asking, well, why is that interesting? Well, that's interesting because we've kind of found out that blood flow shifts in our face when we get stressed. There's one stress system, so we can't really say if this is good stress, happy, excitement arousal, or if it's scared, fearful arousal. We'll have to work that out from context. But we can see a blood flow shift. That's me being relatively calm, even blood flow around my face. But when people get stressed, and this is like acute stressors, the blood flow shifts from the nose and the cheeks, and it gets pushed towards the eyes. So what happens evolutionarily is that if we were in danger, we would want to be more vigilant. And evolution's answer to that was, well, we'll just pay more attention to what's around us. We want everything to be brighter, more vivid. I want to see more accurately in the distance. And so blood goes towards our eyes. Now, as just a side effect of that blood going to the eyes, you get a nasal dip in temperature because it's all moving away and towards the eyes. So this is me when I give talks. Notice the difference. This is a healthy response. It's fine. We should see a change in our stress levels when we get aroused in these ways. So that is totally fine. What's crazy is how fast this works and that you can see it time locked to specific stimuli. So I'm going to show you a video of a participant that we had in the lab. So we did a study that ended in December here. Some of you could have even been participants, I don't know. But this is time-lapsed over about 90 seconds. See if you can see the change in this person's blood flow. So pretty stark, isn't it? So this is in a controlled situation in the lab. We're able to take those measurements, and you can have a think about what you think she was actually doing, because we'll do a quick little game. We're going to do phobia feud. So, fun time. I want you to think about the things that frighten the UK public the most. So, UK surveyed, what are the things that frighten people the most? What would you get that jump scare feeling from? Ah, spiders is on there. It's not first, but it's on there. Shout it out.
Needles.
Oh, needles is a good one. Not on there.
Pigeons.
Pigeons. Think of it, evolutionary sense. What are things that make people scared, like inherently scared? I'm going to give you away the first one. Heights. Heights. Then we have snakes and spiders. Now you can see how these will have stuck with us through evolutionary time. They keep us safe in our environment. What do you think the third one is? Fire? It's not fire? So you might be thinking, why public speaking? We'll talk about that in a second. But just to show you, in a lab situation, now you can guess what that participant was doing. She was doing a bit of public speaking in that video when her nose turned cold. She was doing a condition on the Trier stress test. Has anyone heard of this test before? This is a clinical test that is designed to induce stress in people in an ethical fashion, but it's designed to induce stress. And people fear public speaking almost worse than death, honestly. It's the thing that really puts a scare in people. And so we got people into our lab, and using a thermal camera, we got them to sit down in a chair, very comfy, gave them white noise headphones for 5 to 10 minutes, and then we took a baseline of their face temperature, particularly using the nose as a region of interest. And so this baseline here is their calm, nice baseline of the individuals. They have no idea what they're coming in to do. Then they go into one of the two conditions, and it's counterbalanced across group. And the social stressor is they go into a room, they're sat down, interview style, at a table in front of 4 people, and they're told they've got 2 minutes to prepare a 5-minute talk. Oh, the difference in the nose temperature then dips significantly to almost, 3 degrees centigrade less. The other condition is a pop mental maths quiz, where they're having to subtract numbers from a starting point continuously, and every time they get it wrong, they're told it's wrong, and they have to start over from the beginning. So very much judged on their performance. That also gives them a significant dip from the baseline, but you can see that the social stressor is still a bigger stress for people. Okay, so when we go back to thinking about people's mental health and their well-being, you might think, okay, well, we can just ask them how they're feeling, but actually, the thermal imaging does something more for us. People who are asked how they're feeling, one, may not actually know how they're feeling physiologically. We have a bit of a disconnect. 2, we're primates and we mask. We are evolved to actually hide how we feel because social reputation is super, super important. And we'll talk about that in a second as well. But there's also individuals who cannot tell us how they feel, and those are like the gray apes that we've talked about already. But what about babies? What about many populations of nonverbal humans as well, for the sake of different kinds of conditions? So we do need to think of other methods. But like I said, I probably wouldn't believe you if you told me how you were feeling, because we do mask either intentionally or not intentionally. But the reason is because when we were going through evolutionary time, we weren't the fastest, the strongest, or the largest of primates. And so we wouldn't want to let on to predators if we had a weakness, right? We can see this with some animals, like cats are renowned for not showing when they're in distress, because they don't want to appear weak to predators. Well, we do a similar sort of thing, but not just for predators. We also do it for our own species. We don't want to feel or appear weak in front of our own social partners and our own social groups, and social reputation is hugely important to us. The reason why we think is because through evolutionary time too, ancient man had to rely on cooperative living, right? Because we weren't large and fast and strong, like I said, we needed to live cooperatively in groups to get food, to get shelter, to stay safe from predators. And if you did something that damaged your social reputation and you were ostracized from your group, then it could actually result not just in like a social death, but in a physical death because you were left to fend for yourself. So we really, 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 really care what people think about us. So I thought this would be a good time then to see if we could replicate this Trier's stress test with the audience. Is that something that we could get some potential contributors, volunteers to do? Anybody? Oh, we've got Emily. So what I would say first is this is a lovely little thermal camera, and I'm going to show this to you on the screen. We're going to see how well we're working. That one. There it is. Can you see that? Okay, so should we look at Emily first to see if you can see? Right, so she's already, why is it looking so? Oh, let's restart that. She's already, and she's just going to get more stressed now. Let's try that again. All right, there we go. Okay, so now everybody will likely have a different set point, right? They'll have a different baseline. And what's a healthy baseline and what's not a healthy baseline, we don't know that yet. This is all quite new. But what you want to see is change in the individual over time. So we'd want to see whether or not we can stress Emily so that her nose goes from yellow to blue, right? So we could see Ray as well. Oh, yeah, see, got a little bit greener there. I can guarantee you that mine is as well. Yep. Who's nice and calm? Who's feeling nice and chill? Let's have a look. Shall we have a look? Ah, not as calm as you thought. Yeah. Would you like to stay up here and we'll do the game? Would you like to choose a competitor? And you can see all of yourselves. Look at that. All right, so come a little bit closer. You don't need to see the screen. We can see. Okay, so you can see they both, and this could be that we're just a little cold in here as well. So we've got yellow and yellow with a touch of green there. You can stand a little closer. I can get you both in. So now the game is we ask them, well, we're going to tell them to subtract a number from another number. And the first one to get it shouts it out. If they're wrong, the audience, you need to tell them they're wrong. And then we carry on. So we're going to see best out of 3. Are you ready? Subtract 17 from 2023.
2007.
Wrong.
2005?
Who's first? Who's going to get it?
Oh, 2023.
2023 minus 17.
2000.
We are students, right? Oh, I think he's gone all around it. The audience, do you want to tell them what it is? 6. 2006 minus 17. We're glad they're not doing maths degrees.
1994. Oh, no. 1980.
Oh dear, audience, tell them what it is. 1989. 1989 minus 17.
1962.
Come on, guys. Audience, tell them what it is.
Where am I?
Right, shall we check you again? Well, we've broadened the area slightly. It's not good. Oh, I can see a bit of green coming in there. Yeah, can you see that blue coming in? Right, thank you. Can we give them a round of applause? So you can see definitely that in real time we can track these differences. Where are we at? There we go. And so this is what I'm doing to try to better understand apes who are living in sanctuary settings or living in zoos, because we kind of think that zoo settings are not great for great, generally not enriched enough. Their social circles are not usually big enough to stimulate them in the way that they should be, because they are like us. They want to navigate really complex social worlds. And also those who come into sanctuaries are there because they've already experienced trauma. So they're there because their families have been killed for bushmeat or they've been in the pet trade. So I want to just show you a little bit of how this works. This is us taking thermal into sanctuaries in Africa. This is in Cameroon here. And you can see these satellite cages. These are at the front of the forest enclosures. So they're not in those most of the time. They're in the forests beyond, but they can come up to these satellite cages to get food, medicine, and so forth, or sleep if they want to feel more protected. And this is a Western lowland gorilla here who's showing off to us. Just sitting and chilling. And then this is in Gabon where my PhD students are now, and this is the first time that this chimpanzee group has seen any sort of video or kit like this. So they all came out of the forest really interested to see what is going on. And they've got a whole range of different issues, these chimps. They have illnesses like HIV, TB, and hepatitis. So we're learning about what illness does to the stress and arousal system, as well as whether or not we can help them understand if two individuals are getting along. So using this sort of information to help us understand how stressed they might get when they meet someone new, for example. Here is just a quick image of a Western lowland gorilla called Nona. She was in our Cameroon group, and we were looking at her as a candidate to adopt a new infant that had come into the sanctuary. She had lost her own baby a few years previously to disease, and she looked to be a good candidate because she was very calm and collected. So we did some imaging with her, and we took the imaging when she had her first meeting with the infant. So this was just a visual meeting between the gates. And you can see the nose dip. Can you see how much she dipped there? So we're like, okay, that's okay. She's super excited to see this baby. And we should have. Stress is a good thing for us to keep us on our toes, keep us adjusted and adapting to the changing environment around us. But what we also think is super important, that we self-regulate back down to our own baseline within a reasonable amount of time, so we're not staying up here as super anxious. Well, Nona stayed up there for a very, very long time with that cold nose, so we were a little bit anxious about that, and I'll show you the result of that. And this is Matiba. She is a Western lowland gorilla doing a cognitive game, so this is a cognitive stressor, and you can see that there are many different kinds of stressors that we can have that will give you this nasal dip, some good, some bad, and this is what we're looking into. So let's return to Nona then. This is Nona meeting Nuji for the very, very first time, having their first physical contact. It's very gentle, nice, seems quite positive. This is one of their first real physical meetings. Don't know if you can hear it, but they are laughing. Bit low grunting. But watch the behaviors. Can you see a change there? See, so Nona starts putting up a physical barrier, and then she leaves. And this was the last meeting that they had. You can see Nuji's still very keen, but Nona has absolutely lost patience. She's not ready to take on the responsibilities of a toddler. Not many people are. Anyway, Nuji didn't end up with Nona, and we think that this could have been one of the reasons. She couldn't kind of self-regulate, back down and chill out. She kind of stayed quite anxious, and her tolerance for dealing with the toddler was not great. Nuji subsequently was paired with another foster mom and is getting on beautifully. So that's not a problem. But we're hoping that these kinds of studies can allow us to better understand what's going on under the hood, not just looking at behavior, which can be subjective based on the individual who's recording it, or their knowledge of the other individuals around. So my students who are now in Gabon, looking at those chimps and gorillas, are doing some controlled experiments where they're showing them videos of what we are hoping to elicit a bit of negative stressor, which are snakes and spiders, and these will be ones that are native to that region of Gabon. And then the positive one will be infants of their own species, because we kind of know that when we see infants of our own species, we generally find it pleasurable and sometimes might even have a bit of an oxytocin release. So these are the kinds of things we're doing. And then we're measuring our regions of interest, the nasal area in particular, but also the eyes, because we think the eyes will get hotter and the nose will get colder in relationship. And you can do really, really high frequency measuring. So you're getting absolutely time-locked changes in the blood flow based on the stressors that they're being exposed to, which is so different from capturing cortisol samples that are after the fact. We're also looking at how long it takes individuals to adjust to a new environment. These are two orangutan brothers who came into Port Lim Wild Animal Park here in Kent from a European zoo that decided they didn't want to look after them anymore. So they were 11 and 16-year-old brothers who were very inhibited, very scared, and stayed under a blanket and just were really huddling down. So we did some long-term sort of sampling across about 6 months to see if we could see any change in their baselines across that time. So are these things kind of stuck points, like is my baseline always my baseline, or can we change them? Can we have interventions that allow people to deal with challenges better? And you can see we can do the same thing of the regions of interest and the eyes and the nose and take those samples. And this is about 6 months on. The older brother, the 16-year-old, taking a long time to adapt, still really uncertain. You can still see the broad nasal dip. But the younger brother, and maybe due to the age, settled much better, much faster, more social, more trusting, getting on with the keepers. We also found that even though we couldn't go and take a physical respiration tests of these individuals, with our chimpanzees, you can actually see respiration. So see if you can see the breathing of this very calm individual. So you could easily get some physiological measures using this technology as well. And then here's another example. This is Dixon. So Dixon's in Cameroon also. His keepers described him as having agoraphobia. And why not? I mean, if we share this much DNA with other great apes, they're likely to be prone to the same mental health disorders that we are. Dixon won't leave the satellite cage and go in the forest, even though his friends will come and invite him and try to encourage him to come out. He won't go out. When we put the thermal camera on him, you can see it's not only a dipped nasal area, but it's super broad. And I mean, possibly there are signatures of different mental health conditions, like maybe panic disorder or generalized anxiety disorder that we can see in the different patterns of blood flow. So maybe this is like a chronic nose dip. Right, so as I said, stress isn't bad. We always think about stress as being bad, and you're like, oh, it's a nasal dip, I shouldn't be stressed. But actually, stress and cortisol release and a bit of adrenaline is good for us. It keeps us adapting to changing situations, and we need it to get out of bed in the morning and get our essays done and get our tasks done for the day. But likewise, we need to moderate and come back down and self-regulate and have resilience. And so we were thinking, well, we've been looking at how the stress goes up with the nasal dip, but can we also have interventions that makes the stress come down? And maybe this is like, can we induce a bit of oxytocin release, or are there interventions that can allow us to see changes? And oxytocin struck me as a way to maybe intervene here as a therapeutic intervention, because we know that we release oxytocin when we feel bonded and loved and safe, and that could be from watching infants and puppies and kittens, or it could be because you're bonding with a good friend, or you're in a romantic relationship, for example. And so this is why we actually were showing infants to our chimps and gorillas of their own species to see if it had any effect. And we actually thought that it would kind of be our control, but lo and behold, it seemed to actually have a calming effect. So Julie, she's like a 35-year-old chimpanzee in an ex-biomedical center in Texas. Should have gone to a sanctuary because they've closed all of the medical research on great apes in the States, as they absolutely should have. But some of these apes have chronic illnesses, the same kinds that we get in older years, like congenital heart disease, diabetes, so forth. So they're actually now looked after like they're in an old age home. So they're with their bonded pals, and they have keepers who just give them their meds and look after them and make sure they have a very good quality of life. So I visited them. They don't usually see outsiders, so they're a bit stressy to see me. But this is Julie watching babies. They haven't seen babies in many, many years. This is just an old age home, effectively. And this is about over, I don't know, a couple of minutes. And you can see she's got the nasal dip, but now she's really watching these infants on a screen. She's got a couple of buddies with her as well who are watching from the wings. And you can see over just a couple of minutes of watching that she's coming right back down to a nice even blood flow across the face. So we're really excited about this kind of research. We think it can do a whole bunch of things that we couldn't do previously, but we also know that there's a lot of work to be done. Like, we don't know what is a healthy range of fluctuation when you're stressed. We don't know if some people's set points are much higher and there's huge variability in the population. And we don't know what's a healthy amount of time to come back down to someone's baseline. So this is exactly what we're studying at the minute, but we think that it could have some really strong implications and practical uses, not just in understanding ourselves and other apes, but also in conservation efforts. So helping understand which candidates might be good for fostering infants, or which candidates might be good for potential release into the wild, because we know that they can self-regulate well. We know they're resilient, for example. And making sure that those who don't have the skill sets aren't being released before they're ready, so that we can increase the survival rates. And then, of course, you can't look at one method alone. You have to look at in concert with many other methods as well. And behavioral monitoring is still super, super important. So if this is Dewey, if Dewey had a completely stress-free looking thermal imaging, I would still want to see what the behavior was. And this is Dewey just looking at our equipment. So this is a stress scratch. And this is like when you see a snake, you stamp. So he's very unhappy about, I don't need any thermal imaging to know how Dewey's feeling at the minute. But still, it's really important to have more than one method. And I think it could translate back then to humans. Once we understand it fully in the apes that we're looking at too, we could think about, well, can we monitor people in high stress jobs? What do they look like? What about infants? They can't tell us how they're feeling. We don't know what stresses them out so much. Maybe that would be helpful. And also there are conditions like autism where you have non-verbal populations, and it would be incredibly helpful for us to know when people are feeling overwhelmed and stressed. So this is a group of researchers that I'm currently working with, and even Ophelia, who's come back to have a watch. She did her third-year dissertation on some of these data, and it's, I think, really exciting and fun new work, and I'm looking forward to seeing how it comes around. My PhD students are still in Africa, so they come back in May, and we'll look at that work. If you like this kind of stuff, then I would point you to the new Horizon program that's coming out in the autumn, which is called Secrets of the Brain. And I got the amazing opportunity to film a bunch of this with Jim Al-Khalili. So you can tune into those episodes. Also, because we're here at University of Sussex, Think Again is a new talk series where we look at psychology and neuroscience of different topics with Claudia Hammond, who is our associate honorary professor here. And she's a fantastic broadcaster, but she engages with us for public engagement events. So we put together this talk series. One was last night, but the next one will be probably in November. And then we also have these My Psychology talks now, which are professorial lectures. So it used to be a really big tradition to have a new profs give a lecture as a kind of inaugural sort of thing. And it's not meant to be just about their work, but it's meant to be about their journey through academia. And we've got Jessica Horst doing one in April, and there's a big backlog. So it doesn't mean that these people just got their appointments. So for example, I'm doing mine in May. I was made professor in 2021 or 22 during lockdown, so I never got to do one. And so we're going through all the backlog, and I'll do one as well. And those are just in Fulton A here, so that should be really lovely. And if you really like science and you're coming to Glastonbury, I'm running the science area there this year, and do come and see us. Or if you're going to be about and you want to help out or do something on our laboratory stage, come and find me. And thank you so much for your attention.